Good evening and hello everyone. We are going to commence this month webinar conducted by Institute of Physics Sri Lanka. I'm Shanta Gamage, the president of IPSL. The main objective of the IPSL is to promote and advance the science of physics in Sri Lanka. Before introducing our resource person today, I would like to warmly welcome all the participants of this webinar. Thank you for making the time to watch our seminar today also. So today we have invited Dr. Dilmini Wijesinghe, a postdoctoral research associate at Mark and Mary Steve Neuroimaging and Infantry Institute, California. So before formally beginning the presentation, let me call upon Dr. Vasanthi De Silva, Joint Secretary of IPSL, to introduce today's resource person. Thank you very much, and over to Dr. Vasanthi.
Thank you, sir. So before we start the webinar today, I would like to introduce our guest speaker. Dr. Dilmini Wijesinghe is a postdoctoral scholar and a research associate at Mark and Mary Stevens Neuroimaging and Informatics Institute, Los Angeles, California. She obtained her bachelor's degree in physics from University of Sri Javadhanapura and her PhD in medical physics from Auckland University, Michigan. Prior to joining INI, Dr. Wijesinghe received her postdoctoral training in the Department of Biomedical Engineering at Duke University. She is a member of Psi Sigma Xi Scientific Honor Society. Dr. Wijesinghe's research interests are focused on mathematical and computational modeling of biological phenomena, neuroimaging, complexity analysis of fMRI, and deep brain simulation. So I would like to welcome Dr. Dilmini Wijesinghe. Over to you, Dr. Dilmini.
Thank you so much. And these are my peers. Actually, I studied in the same department, and Dr. Shanta is one of my professors who taught me. So I'm really glad to be here, and thank you so much for inviting me. I'll start the presentation. I hope all of you are ready. Hope you all can see the screen.
Yes, we can.
Okay. I selected a couple of examples for today's presentation. So I worked on mathematical modeling of biological phenomena during my PhD and also during my postdoctoral studies. So today I selected the applications of from the biological phenomena, for example, for the biological phenomena, mechanotransduction and the deep brain stimulation. So I will go more descriptive examples for mechanotransduction, but for deep brain stimulation, I don't think with the time allocated, I will not have enough time. So I will be mostly giving it as an example and also applications of physics and mathematics in deep brain stimulation. So our human body reacts to 3 different types of signals. So one is chemical signals, and also electrical signals, and also mechanical signals. So we have 3 different words for each of these type of signal. One is the mechanotransduction for chemical signals, and reaction for chemical signals we call chemotransduction, reaction for mechanical signals we call as mechanotransduction, and reaction for electrical signals we call as electrotransduction. So what I am interested on is mechanotransduction. So that's basically the idea of when we give a mechanical stimulus to the human body, and the cell receptors sense this mechanical stimulus and convert it into a biochemical signal. And then eventually we see a response through the nervous system. So from this whole, these steps, this procedure, I'm not interested on biochemical signals. So what I am interested on this connection over here, where how the cell receptors sense the mechanical stimuli. So there are many different models to predict this one. And actually in medicine, the world of medicine and biology, we really don't know how cell receptors sense a mechanical stimuli. There are a couple of hypotheses, but none of them actually being proven, but some are effective, but it's really hard to test how cell receptors sense the mechanical stimulus. So before I begin the talk, I would like to give an overview of the scale that I'm talking on. So human heart is about the size of your fist, and the cardiac tissue is somewhere around 1 millimeter, and cardiac tissue fibers are roughly 100 micrometers long. And around the cell, there is the cell membrane, and the width of the cell membrane is somewhere around 10 nanometers. So I apply a model called mechanical bidomain model to understand this mechanotransduction. So if we zoom into the cell membrane, and the cell membrane, there is intracellular and extracellular spaces. So the intracellular means inside the cell, extracellular means outside the cell. It's a little bit complex because there are so many proteins and other factors that contribute to make it as an extracellular matrix, and also same goes for the cytoskeleton. So in the cell membrane, there are proteins. There are different kinds of protein that are responsible to do different kinds of work to make it our metabolism work. So there's one particular protein called as integrin. This is a mechanosensitive protein, and it can sense the stimuli. But the problem is how it senses, that's we don't know. So according to the mechanical bidomain model, the bidomain, the word bidomain means intracellular and the extracellular, the two domains. And all the other models are called as monodomain models. So basically they are either fluid-based or more like a weighted average of the two spaces. So to begin our derivation, first I would like to indicate the displacement of the intracellular and extracellular as U and W are both vectors. So the key hypothesis of the model is that the difference of the displacement of these two spaces triggers a force on the integrins, thereby activates the mechanotransduction in the cells. So as I said before, there is electrical signals too. So there is electrical bidomain model, and the mechanical bidomain model is structure-wise very similar to the electrical bidomain model. And the electrical bidomain model was discovered in somewhere in the 1980s, and it is very beneficial to all of us, because current modern medicine, actually, we are experiencing the benefits of finding the electrical bidomain model and also the electrical model of the human body, because we have pacemakers, EEG signals, ECGs recordings. These are all thanks to the electrical model of the cells. So in the electrical model, the extracellular and the intracellular spaces, we represent using a set of resistors really connected. And also there are ion channels, that is the coupling between the two spaces. So the ion channels are represented using a parallelly connected resistor and a capacitor. Whereas in the mechanical bidomain model, all of these are springs, and we have, instead of ion channels, integrins, the protein and the mechanosensitive protein. And all these springs obeys the Hooke's law, the basic spring, the Hooke's law that we all learned somewhere in grade 8-9. So the beauty of using this kind of a model is that the building block of the model is a cell, which is microscopic scale. But when we can extend this model to a 2 dimensions or 3 dimensions to talk about macroscopic properties as tissue properties. So in the mechanical biodomain model, this is the two-dimensional version. So we can have the extracellular space as a mesh of resistors, springs, and also intracellular space as mesh of springs. And we can sandwich the integrants in between, and we get a two-dimensional version of the mechanical biodomain model. And not only structural-wise, in the electrical and the mechanical biodomain model, there are other similarities. So the key hypothesis of the electrical biodomain model is the difference of the potential between the extracellular and the intracellular spaces activates the ion channels, whereas in the mechanical biodomain model, difference of the displacement activates the integrants. And also, there is anisotropy. So it's being tested and proven that cardiac cells and all the other cells in the human body, they are anisotropic when they are passing electrical signals. So that means, I will describe this later actually in more detail. Also, even though it's hard to prove that concept in the mechanical signals, but we assume that mechanical signals, the cardiac tissue should react in a way that the cells are anisotropic, because one reason is that the way that the signals pass from the inside of the cell to the outside of the cell, and also inside from a cell to another cell, where desmosomes is a little bit different. It cannot be the same way the force and the mechanical modulus can work. So this is the two-dimensional version, and this is the three-dimensional extended version of the mechanical biodomain model. So it's more like two cubes coupled together. So I applied the mechanical biodomain model to different, to observe different phenomena. And from here, I would like to pick up one, which is trying to bring the anisotropy to the cardiac tissue and then test the indentation. Because we build a model, we start with a very simple model, and we wanted to implement the model eventually, so then we can bring the actual properties of what we are observing, and then eventually model become more realistic. So now my goal was to bring anisotropy and implement the model for 3 dimensions. So the word isotropy means direction dependent. Whatever the material is, it doesn't when the signals are passing through, or any property that we are observing, it's direction independent, and anisotropy means direction dependent. So there is another hypothesis that we are pursuing through this derivation, that is cardiac tissue is incompressible because tissue is mostly consists of water. And to bring an anisotropy, we create an unequal anisotropy ratio. So this ratio is the easiest way to describe about this one is using the properties of an ellipse. Anybody who worked in MRI physics will be probably familiar with using an ellipsoid to describe the anisotropy. So eccentricity, we're going to consider as how close to being isotropic, and semi-major axis as mechanical modulus along the fiber direction, and semi-minor axis as mechanical modulus perpendicular to the fiber direction. So as an example for the isotropic case, we can take two circles for intracellular and the extracellular space, where the eccentricity, because it's circle, eccentricity is 0, and then we can take the ratio of these two. If the ratios are equal, then and eccentricity is 0, then it's isotropic, and ratios are not equal, but eccentricity is equal, then it's equal anisotropy. When eccentricity is different and also the ratios are different, that is unequal anisotropy. So we start our derivation by using the stress-strain tensors. So the tau represents the stress and the epsilon terms represents the strains. And the I-side density vector P and Q are the pressure terms. So anywhere that you see a subscript I, that means intracellular space. Anywhere that you see a subscript E, that is extracellular space. Also here, the gamma I and gamma E are the gamma I is the mechanical modulus, which is the anisotropic property of the anisotropic property of the gamma I represent an isotropic property of the intracellular space, and gamma E represents the anisotropic property of the extracellular space. And mu and nu are the mechanical modulus of the intracellular and extracellular spaces. Other than the tensors of stress and strain, we use three-dimensional equations of the mechanical equilibrium. Here, the K represents the coupling constant. And also, we can use the mechanical bidomain to model, observe the monodomain and bidomain displacements. So according to our hypothesis, the bidomain displacement, the magnitude of the bidomain displacement is the one that contributes to activate the mechanotransduction. As I said before, all the other models are monodomain models. So the monodomain models hypothesizes that stresses and strains activates the integrins, but not exactly how the integrins sense the stresses and strains is still not being answered in monodomain models. So in the monodomain model, we consider the two spaces as a weighted average, whereas the bidomain displacement, it's more like a difference of the displacement. So to plan the tissue indentation, we consider the box, which is the very cube shape, very basic shape that we can consider in three-dimensional space. And then there are different types of indenters. So in this case, I tested this with a pyramidal shape indenter, and we can use it spherical indenters. So you can change the indenter. So the tissue indentation is a very well and widely known technique to understand. It's an experiment that mostly conduct to understand the mechanical properties of tissues in the human body. So due to the symmetry, we only have to mathematically implement 1/8 of the cube. So you can see a faint shadow of the indenter at the bottom surface too. So then you can guess the symmetry. So to implement these analytical equations in mathematical, numerical methods. So in numerical methods, I used the finite difference method. Of course, there's finite element method too, but it's a cube shape, so we don't need to make it more complicated. So for now, I'm explaining how we use the finite difference method. So in here, the filled voxels represents the cardiac tissue and empty voxels represent the phantom nodes. So if you're familiar with the finite difference versus finite element models, finite difference model, the biggest challenges are calculating corner nodes and the boundaries because of the instability that it brings. So for that, these phantom nodes are really important, and it makes it more easier to calculate mostly unstable nodes. So we tested this for multiple boundary conditions. So both intracellular and extracellular space pressed downward, on the extracellular space being pressed downward. So there are some experimental evidence also some of these boundary conditions, but especially the one in the case 2, which I'm not going to describe. If you're interested, we can talk about it. And these are the equations that we derived. So in here, as you can see, the terms that has M represents monodomain, terms that has B represents bidomain terms. So when we change the isotropic and anisotropy ratio, we can get isotropic tissue or equal anisotropy or unequal anisotropy, depending on how we change the value of the ratio. So when we change the value of the ratio, some of these terms goes to 0. So we can get some coupled situations of the equations and uncoupled situations. So when it's coupled, it's really hard to solve it in numerical methods, but we were able to establish the numerical methods is using the finite difference method. So as you can see that the top surface is being pressed downward in the figure. That is the extracellular space. And when we try to observe this mechanotransduction, which is actually derived using the difference of the bidomain displacement, you can see from the top surface to more towards when we go deeper down in the layers, how the signal is being spread through the tissue. So this is, again, because of the symmetry, this is 1/8 of the cue. So the next one I'm talking about is deep brain stimulation. So this is more like an application, and where, again, mathematical modeling and physics that we can apply in medicine. So deep brain stimulation is a surgical therapy that basically most commonly being applied to people that has Parkinson's disease or tremor, with the age that some people has a tremor that they cannot hold a cup or their own food, and they have this constant tremor in their hands. So to avoid these kind of diseases, deep brain stimulation is a very effective treatment therapy, and it's widely known therapy for movement disorders, basically. But I did not work on applying deep brain stimulation for movement disorders. Instead, I applied my projects are actually applying deep brain stimulation for clinical depression. So now, if you're probably be confused why we use that kind of a invasive surgical experiment for clinical depression. This is actually currently at experimental level. This is not widely known treatment therapy yet. So, but still, people do undergo this treatment therapy only if they give the consent and they agree, and all sorts of other conditions. So in clinical depression, there are different levels of clinical depression, mild and to sadness to severe or persistent depression. But the clinical depression in normal depression, we have different kinds of levels, but when it comes to clinical depression, it's very severe than the normal forms of the depression or the normal levels of the depression. And also it's known as the major depression or the major depressive order and disorder, because it's persistent, but also it affects a lot of the function of the body. And to diagnose the clinical depression, physicians basically use different tests, feedback tests, and also signs and symptom observations. So why we are interested in bringing the deep brain stimulation to clinical depression is because it slowed a person's thinking, speaking, and body movements. Patients that undergo depression over a long period of time and also have the symptoms of clinical depression. Clinical depression, actually, I wanted to highlight, it's not like, it does not basically occur, it can be, but it's not mainly occur because of the loss of loved ones or because of a disease like a thyroid disease. But this can be, there will be very big reasons than these kind of reasons. Basically can be due to the evolution of the brain, because someone's brain can be somewhere like at age of 14, which it has to be developed to some certain level. But if it's not developed to that certain level, then that person can suffer from clinical depression. So there is no relevance of age to someone to suffer from the clinical depression. So when someone is going through the clinical depression, eventually over the time, you'll observe that they won't be able to get up from the bed, or they don't like to do any daily day-to-day work. They're not enthusiastic to get up and go to work. So eventually their body movement is going to be very limited, and they're going to reject to do any sorts of basic movements eventually. So that's why we are bringing the deep brain stimulation. So deep brain stimulation is that there are 2 electrodes. Actually, we call it a lead. Inside lead, there are actually 4 electrodes each side. And there are 2 DBS electrodes implanted basically in the brain for each side of the brain. And there is an implantable pulse generator that's actually implanted on the shoulder. And so I would like to a little bit talk about the leads. So there are different kinds of leads, There are two companies that are major companies in the United States that develop these leads. One is Medtronic, one is Abbott. So the Medtronic is the lead that we used in our experiment, and those are circular electrodes. So the electric field is evenly distributed around the area, whereas Abbott, which is Abbott electrode leads are very modern, and very recently they developed these asymmetrical electrodes in the leads. So when the electrodes are asymmetrical, the electric field can be asymmetrical too. When the electric field is asymmetrical, depending on each side of this electrode, we can control the intensity of our electric field. So it's more advanced kind of leads. So programming these leads mathematically is really complicated. So what happens, this is actually a collaboration with Mount Sinai Hospital in New York and the Dr. Helen Mayberg's group. Dr. Helen Mayberg is the first person in the United States to suggest deep brain stimulation for clinical depression. So they implant these electrodes in patients' brains, and they take MRI scans before and after, and CT scan and other bunch of observations from the patient. And they send all these details to our lab at Duke. And so from the MRI images, I was able to extract the pathways and recreate the white matter fiber tracks that we were planning to observe. So we are interested in three particular fiber tracks in the brain. One is called forceps minor cingulum bundle left, and the other one is cingulum bundle right, because they are basically responsible for creating sad thoughts. So our goal is to implant the electrodes, give the pulses from the IPJ to suppress the neuronal activity in this region. Instead of activating it, we are trying to suppress the neuronal activity. So once I extract these pathways in the brain, we program that one to make the distances from each coordinate to be equal to the Ranvier nodes. And then we can also find the electrode locations, and we can do an activation map of these pathways. There are other softwares that we can use like Neuron, but this one is basically from the scratch program from MATLAB. So you can program to activate different regions in the brain using these kind of maps of pathways in neuronal pathways in the brain. So this is the basic idea of the experiment. So they do a once after the surgery, after a few weeks, maybe 4 weeks to 8 weeks after, the patient comes back to the hospital, and then they take a CT scan, and which we can see the lead location. So we take that coordinates and bring it to the DTI space, and eventually the T1 space, which is the coordinate space where the patient's head is. So this, if you're familiar with MRI physics, you're probably familiar with DTI and all these different spaces, coordinate spaces that we are talking about. But this otherwise just consider this as different coordinate spaces. So so then once we reconstruct these fiber tracks, and we use different kinds of models. One is called the driving force predictor model. It's another mathematical model, and also lead in the box model. But this lead in the box model is a very simple mathematical model of the DBS electrode. So instead of the head, we take a box and a lead inside, and we can use numerical methods. Again, it can be finite difference or finite element. In this particular case, we use finite element model. So finite element model to model the electric field. There's actually, the lead in the box model is the very simplest version, but then we can implement that into a realistic head model of, we can use the finite element model to model a realistic head, and then there's a lead inside, and then thereby we can calculate the electric field. Then from that two models, we calculate the thresholds, potentials, and also what is the best optimized settings for the electrodes. Can be monopolar, dipolar, or quadpole. So because basically clinicians have always used the we call C1, C2, C3, and C4. C2 and C1 and C2 and C3 electrodes, the middle electrodes. They only rely on the patient's feedback to adjust this, to choose which electrode is the best. But we're trying to give them a reason, a very good reason to activate or choose the electrode, and also what is the best place to place the electrode to in order to activate or deactivate majority of the pathways. So that's what the optimization means in here in this particular case. So we use this energy optimized settings, and also we are planning to save the battery, because less energy means more time. Patient doesn't need to come to change the battery. So basically, one of these batteries last for like 10 years. So then another few weeks passed, let's say from the 8th week to 26th week, patient is going through a recovery process. So when they're going through the recovery process, the scar around the DBS electrodes heals. When it heals, impedance around the DBS lead is changing. So when the impedance changes, we can change our threshold and potential calculations according to that too. So they do send those measurements to us too. So once we calculate all these settings, we send it back to the hospital. And in the hospital, patients comes back, and for like 10 minutes of the time, they change the IPG settings to the settings that we send, and also place an EEG cap in the brain. So EEG cap looks like this. So there are, in this EEG cap, there are 257 electrode sensors, and then they record an EEG signal. So that EEG signal is a high density EEG signal. And if we put them, all of those signals together, it's going to look like this for one particular patient. And that big spike represents the stimulus, the given stimulus. And so if you zoom in, the signal going to be something looks like this. And our goal, so we are particularly interested on 10 sensors only. And we trying to see, reduce the noise or extract these signals and try to observe some kind of a neuronal signal. So we use different types of techniques, different types of filters. One of the main filter that we use to extract these features is called Savitsky-Golay filter. So we do extract these features to observe if we see any kind of a clustering or pattern of these signals. And so these are basically the applications where the physics and mathematics are going to help the clinicians. And also currently I'm working on, so these are all that type of signals that we are trying to observe with different filters and removing the noise signals from these electrodes. So another application that I'm currently working on is MRI images has noise signals. So the noise basically represents chaos. So we are using entropy to model these noise signals. So we use different types of entropy tests, like complexity, sample entropy, the different kinds of entropy tests. Some we design and implement mathematically and then apply them in and develop softwares to test those signals in MRI. And also we can do machine learning techniques and things we can apply to denoise these kind of signals like EEG signals. Yeah, so those are the applications I would like to bring up, and hope this presentation is helpful and give some insights about medical physics applications. Thank you.
So thank you very much, Dr. Dilmini. It's a very interesting presentation and it's an impressive work. If anyone would like to ask any question, please feel free to ask. So I have one question about the implantable pulse generator. So is it sensitive to the external magnetic field or electric field? How about the sensitivity of that?
Inside the body or outside?
Outside, yeah, I mean.
No, it's really low actually, sensitivity, because we can actually conduct MRI scans too. But as a precautions, we do not. In our case, we do not do an MRI scan to when patient has an IPG, but we do a CT scan. But there are situations that they do use somewhere like 3 Tesla MRI scans, which is very safe to use.
But in the case of pacemaker, it's a little bit sensitive to
yeah, pacemakers are sensitive to the external magnetic field. Yes,
yeah.
True. You had to remove the pacemaker if you're going for a scanner.
Yeah. Right. Any other questions or clarifications? Actually, I have one more. That is,
sorry. It's all right.
Yeah, it's very interesting about that modeling part. So in seismology also we use that type of modeling. So, but you apply isotopic case, but how do we assume that this is isotopic when we model that? Because it not actually a liquid type one. So it's possible to assume as isotopic or?
What do you mean by liquid? You said liquid in time?
Yeah, I mean, when, yeah.
The one thing is that this is a very slow process, even though the heart, for us, when it feels like heartbeats faster, and then for that one, it's large stresses and strains going to work when for a beating heart, like day-to-day mechanics that applies. This one particularly, which is the model that I showed, is basically focused on small stresses and strains. So the reason that we focused on smaller stresses and strains is because there are abnormal tissue growth happening in human body. It can be in the heart, or it can be any other organs. So the reason for this abnormal tissue growth is something unknown, especially there are diseases like cardiac hypertrophy. So, so this is a very long process, the time-wise. So we do not add the time span, the time as a property to the model too, as one of the things that I wanted to mention. So, and also the isotropy comes into the picture because we consider that there is no difference the way that signal passes from inside of the cell to the outside, or from within the cells, because there are two pathways. So this pathway is the one that we are calling isotropic and anisotropic. Does that answer the question? Hello?
Yeah, I got it. Thank you very much. Yeah. And any other questions? Any more questions? So if no more questions, let me thank Dr. Dilmini for his interesting presentation. And I would like to invite Dr. Rahubad, Joint Secretary of IPSL, for vote of thanks. Over to Dr. Rahubad.
Thank you, Professor. Suddenly, I got the current failure and I tried to manage it. Dr. Dilmini Wijesinghe has successfully conducted the lecture series in mathematical modeling of biomedical phenomena and Mechnotransduction and deep brain simulation. That was a very supportive and helpful moment in upgrading our knowledge and understanding regarding the Mechnotransduction and deep brain simulation. On behalf of our President, Professor Shantha, Council members, and all the IPSL members, I would like to convey my sincere gratitude to Dr. Dilmini Wijesinghe. Dr. Dilmini Wijesinghe, I wish you all the very best. Thank you, my friends who worked to make this event success. Thank you, audience, for your participation. Good evening and have a nice weekend.
So thank you very much.